All right, this is going to be the review for Chapter 16 on DNA structure and replication. This first slide is depicting the structure of DNA with the double helix on the right and what the base pairs look like lined up inside the DNA strand. So first thing you got to take note of is that the DNA backbone is made up of sugar phosphate molecules. Each sugar and phosphate is part of one nucleotide, but the phosphate always connects to the 3' carbon and the 5' carbon of the bottom nucleotide. So the way this works is the sugar phosphate backbone just keeps connecting 3 to 5, 3 to 5, 5's right there, 3 to 5. And as you go down this sugar phosphate backbone, that's what makes up the outside part of the double helix. Now, as you can remember from when we talked about nucleotides and organic molecules, the 4 nucleotide nitrogenous bases were adenine, thymine, cytosine, and guanine. And the way these lined up was that A always matched up with T and only had 2 hydrogen bonds in between each of them, and G always bonded up with C and always had 3 hydrogen bonds between them. Those base pairs always consisted of 1 purine, which are the 2 smaller ones, which is A and C. So we'll do this here, A and C. And actually, I was mistaken though. The A and C are pyrimidines, not purines. And let's do a different color for the other ones. Thymine and, wait a minute. Hold on. I believe it's thymine and guanine that are purines. All right. So as these two, as those line up, they always actually have to have the 3 helical structures in between them, so that the DNA sequence stays uniform throughout the whole DNA strand, and that it's not bubbling out or dimpling in if there are less molecules that need to be there. So the A always matches up with the T, and the G always matches up with the C. Last thing we'll talk about with this picture here is the DNA runs from 5' to 3' end, and DNA strands are anti-parallel. And what that means is that even though they are parallel to each other, one runs in one direction, and the other one, like this one on the right here, runs in the 3' to 5' end. What do I mean by that? Well, what we're referring to when we say 3' end is that the top carbon, the carbon on the top of this strand on the right here, is the 3' carbon. If you go around the whole ribose sugar here, you have the 1, 2, 3, 4, and 5. So if the 3' end is up, that's the 3' end, and it's running in the 3' to 5' direction. Again, on the left, it's the opposite way. And remember from lecture that DNA can only grow in the 5' to 3' direction. 5' to 3' direction. So without DNA growing in that sequence, you can't actually make new DNA strands. So throughout this entire diagram, the overall picture is, again, this double helix. Watson and Crick, with the help of Rosalind Franklin and Wilkins, discovered this structure. And yeah, that's the basic overview of DNA structure. So this next picture we're going to look at is DNA replication. And DNA replication contains a few different proteins. The first protein here is helicase. And remember what helicase did was it separated the 2 DNA strands. When you want to replicate a strand of DNA, the first thing you got to do is separate the 2 strands and then hold them open. Okay, so the helicase is the protein that's separating them, and the single-stranded DNA binding proteins, which are these black dots right here, hold the DNA strand open so that the DNA just doesn't close in on itself right after helicase opens it up. Following the helicase, we have Rna Primase, or just primase. And what primase does is it lays down a little tiny strip of DNA, which allows it to, actually it's a strand of RNA, which is almost like a giant arrow pointing at which DNA strand needs to get copied. And DNA polymerase, which is the actual enzyme that copies the DNA, doesn't really know where to land, and it needs the help of RNA primase to know where to land and start copying. So RNA primase is the protein that actually lands on whichever gene needs to be copied or whichever strand needs to be copied, lays down that primer, and then DNA polymerase can do its job. In this picture, it depicts DNA polymerase 3. There's different numbers of DNA polymerases. You don't need to know that right now. But one thing you do have to know is how this strand then is copied. So as the DNA strand opens up, as helicase opens it up and the binding proteins keep it open, DNA polymerase is going to land right ahead of the RNA primer, so right about here, and it's going to start copying the sequence that is following the RNA primer. On the 3' to 5' end, the DNA polymerase can go straight through. This is known as the leading strand. And it's the leading strand because as DNA polymerase copies it, it's usually way ahead of the other strand, which is the lagging strand. So because it's able to copy the DNA from 5' to 3', it doesn't have to stop or halt its copying in any way. It just keeps copying and copying and copying until it reaches the end. On the lagging strand, however, the lagging strand is termed that because it's always a little slower. And what the problem with this strand is that it's initially, as it opens up, already in a 5' to 3' direction. And again, as you know, DNA can only grow in a 5' to 3' direction, and since DNA is anti-parallel, the DNA strand has to grow in this direction somehow. So as the DNA helicase opens up the DNA strand in this direction, the RNA primer keeps having to go backwards and add little RNA strips to copy this tiny segment of DNA. What these tiny segments of DNA are called are Okazaki fragments, named after the scientists who discovered them. And after all of these little tiny fragments are copied, the enzyme ligase comes by and glues them all together. So those are the proteins that are at work in DNA replication. And that's the overview for that. Now, if one other protein that we haven't talked about is Rna Nuclease. And what Rna Nuclease actually does is, let's say there was some error in your DNA code. Again, remember, DNA polymerase is not a 100% accurate protein. It's going to make a few mistakes here and there, not many, but when it does, and let's say it places a T next to another T, this sequence is then going to dimple in, and it's going to create an altered structure of DNA. And following that, Rna Nuclease, which is a protein that fixes DNA, comes by, cuts out the segment of DNA, and allows DNA polymerase to then come back in and add the appropriate nucleotide. So through this whole process of fixing DNA, DNA can actually go back and really correct itself, and most of the mistakes that occur in DNA replication won't keep happening over and over again if they go unchecked. So that's DNA fixing. And then the last thing we're going to talk about is telomeres and telomerase. Again, when DNA gets replicated, DNA first has to land down a primer done by primase, and then the DNA polymerase comes and starts its DNA sequence right after the primer. Now, after the whole entire DNA replication has been completed, DNA polymerase can go back through the chain and convert all of those RNA primers into DNA. However, it can't actually convert the first primer into DNA because it has nowhere to add bases to. Remember, bases can only be added to a 3' growing end. If there's no 3' end to add bases to, oh, that's a D, not a G, then the DNA strand can't grow at all. So what actually ends up happening is that every single time you copy DNA, a little tiny segment where the RNA primer was on gets chopped off the new DNA strand, and that's ultimately what telomeres are for, is so that you have actual DNA to be allowed to chop off and it really doesn't affect any of your genes. Now, what telomerase does, which is evident in some cancer cells, is that it's a special enzyme. Telomerase is a special enzyme that can come back in and add DNA retroactively to fill in these gaps. And that's, how telomerase actually allows cancer cells to live forever, because their DNA strands never run out and they keep copying over and over again forever and ever. We don't have that enzyme. However, some cancer cells do. So that is really the crux of how cancer cells keep living, and why our cells end up dying, because we just run out of telomeres to chop off. So that's going to be the review for Chapter 16, and we'll see you next time.